What? Just want you to take me to my mail. Chapter 2. I don't know. I'm just assuming people aren't going to class just because it's raining, which is not an excuse not to come to class, just to be clear. It's just raining. So I feel like, hopefully people just running late because traffic, because the rain, there is a lot of rain on the road. There is a lot of rain on the road. Yeah, for sure. So I'm assuming that's why people are running a little bit late. So we'll get started here in a couple minutes. But I'm hoping that people will at least show up, even if they're a little bit late. There.
No, I never really which one was it. So I wrote about that was
Anybody have questions about anything before we get started? I will probably have everything graded by the end of today. In case you guys can't tell, I usually stay on top of that pretty good. I did actually read everybody's concept maps, everybody's lab submissions, everything like that. And so I am grading accordingly. So I'm not saying it was anybody in this class, but there's some people that definitely submitted the concept maps, and I don't know if they thought I wasn't going to read them or that I wouldn't notice spelling mistakes or things like that, but I do grade for stuff like that, and it is in the rubric, so that you guys are aware of that going forward. I tried to be a little lenient this time. If you got a comment from me about, again, this is the first week and we had 4 due, and I know it was a lot, so I was a little bit lenient with that stuff, but I'm not saying I'm gonna be super harsh moving forward. It's not meant to be a super harsh exercise, but I am gonna call you out if you're missing major concepts, right? For the concept maps, if you guys are having issues like figuring out what needs to be on your concept map and stuff like that, focus on the learning objectives. Start there, and then build out your learning objectives as you need to make sure that you're covering everything so that it's an appropriate study tool for you. But I'm at least looking for the learning objectives. So if the learning objectives said that you needed to define the terms relating to, flagella arrangements, and you just listed the names of the flagella, it's not enough. And so you might have gotten dinged for stuff like that on your concept map. So keep that in mind moving forward. Like I said, I wasn't super picky about things when I was grading them, but if I saw it, I did ding it, right? A lot of people forgot to put their reflection questions, and I mean a lot of people forgot to put the reflection questions. So I am giving you today. So if I have notified you that you are missing a reflection question and you got 9 out of 10 points on your concept map, get that reflection question in either your comments or submit it with resubmit your assignment, and it'll mark it as late. If you resubmit it, don't worry about it. I'll take off the late penalty for it. So just make sure that your reflection question is there so you can get your 10 out of 10. You don't want to miss out on that on these early chapters. So keep up that good work with that. I was actually pretty impressed by you guys' work on that. I know some of you guys will submit it and it'll have that. Some people, if you submit like a picture of like a handwritten one, the thing that identifies plagiarism, it can't like read the pictures of like handwritten ones, but I'm less worried about handwritten ones, right? But if you do submit an outline or a PDF version of yours and it gives you like 25%, similar to somebody else's, don't worry about it. Don't worry about it. Just, of course, it's going to be similar to other people's stuff, right? It should have the same info on it. Yeah.
I didn't get to finish the chapter 4 one.
Uh-huh.
I was planning on like submitting it after class today.
It'll be a late penalty.
Okay.
Yes.
How much did that take off?
25% a day.
Okay.
Yes. So on my class, late penalties are 25% a day. And you're welcome. Like if you guys didn't get it in on time, you're welcome to submit it late and take the 25%. It's better than not getting the credit, right? For sure. So if that ever happens, or you forget to do a quiz or something like that, still get it done, but take the late penalty on that. So, yes. So any other questions? Also keep in mind, right? Your lowest concept maps, your lowest 2 concept maps do get dropped. So if you didn't do one or you didn't get to it, you can just take a ding on it. Or if you want to save those for later, you can save those for later. But that's on you guys. So most people got them in relatively on time. If you submitted it to me before like 10:20 last night, I got it graded. But if you submitted it after 10:20, and I'm saying like about 60 people, I had 60 things I need to grade still. So it's about 60 things were submitted after 10:20 last night. So yes, so we also are starting to learn that the concept maps aren't something you can just knock out in like 10 minutes, right? It's something that you have to put a little bit of thought and organization into. So waiting until the night, like waiting until 9, 10 p.m. the night before 4 of them are due is not going to do it. No, normally we 2. Normally it's 2. Or this week 1 was different because we didn't have week 1 and 3. That's why I said I'll drop the 2 lowest. So if you didn't want to do 2 of them, only do 2 of them. But that was on you guys' choice on how you want to use that. But yes, so normally it's 2. There's 1 week where you will have 3, but they are shorter. Like chapter 25 is the one, like it'll be 9, 25, and 10. 9 and 25, 25 is very, very short. So that should have like three bubbles around it, and you'll probably cover everything. So yeah, so keep that in mind. Some weeks we won't have any. If we have an exam that week, we don't have concept maps due that week. You just have to worry about the exam, right? And then like next week is Labor Day. So we don't have lecture that week, so you don't have concept maps that week. So it's only like when we have lectures that have concept maps. So, so some weeks you'll have none. This was a rough one, and most of us got our 9 on time. So I'm actually pretty proud of you guys for getting them done at the rate that you did get them done. Again, it's on purpose. They're designed to be a little bit more work on purpose because, like, technically, and this is the thing that I brought up in my online class, I'll say to you guys, technically, this is the type of studying you should be doing for your tests for all of your classes. So this is like when your professors are like, study for your test, they don't mean do Quizlets and flashcards. They mean go through your notes, summarize them, reword them, and organize them and see how they make sense to you. So hopefully this will be a useful tool for you guys. It'll help with your studying because now you have a sheet to look at to remind you guys of all the stuff you're supposed to know when you guys are studying. And that's a great review mechanism for whenever we do the exams. We'll have exams pretty much every month, pretty much, like not in August, but starting in September, pretty much every month we'll have an exam. So that'll keep you guys on top of that stuff. All right, so we're gonna do our Kahoot quiz. Everybody make sure you're signed in, and we're gonna get going. And I'm gonna call on people today, right? This is when we're doing our normal thing. It's our first time getting into that procedure. So I will be calling on people. If people aren't here, so that you guys know, so that if you're late or something like that, I know what the procedure is. If you're not here when I call on you, I will come back at the end of class, after I've gone through everybody, to those people that weren't here or whatever. That's your chance to get your score on that. Otherwise, if you're not here, you get 0 for those. You get the 2 lowest scores of this dropped as well. So you can miss 2 days of class if you were planning. Okay. All right, so let's go ahead and get going with the Kahoot. Everybody ready? Anybody still need to sign in? We're going to go. All right, first question. Use your concept maps. Hopefully we're ready for chapter 2. It's our chemistry review stuff. First question, which type of chemical bonds involve sharing electrons? All righty, so most of us got this one. Covalent bonds are sharing electrons. Is Isabella here? Okay, yeah, it's gonna be you. So tell me why ionic is the wrong answer. Sorry, you can look at your notes. You got a minute. So we saw that covalent is where we share electrons. Why is ionic? So we know it's not sharing electrons. Do you remember what happens with the electrons? Anybody else know? Anybody want to help her out? Yes.
And they're transferred.
Transferred, right, or donated, right? So in order to get your credit, I'm going to give you a little chance here. Just go ahead and repeat why that was. So if we had somebody else answer it, let's see if you can repeat that answer. Because we're transferring, right? Okay, great. So that's fine. That's getting us to talk about this stuff, right? All right, next question, let's move on. All righty, hydrogen bonds. Tell me about hydrogen bonds. Be sure you read the choices and understand what the choices are telling you. All righty, so we're a little confused about this one, it seems. So this is going to be a little tough. So the answer was it holds water molecules to one another. Now that might not be the definition of a hydrogen bond. That's not maybe how we would define a hydrogen bond, but this is an application situation of the hydrogen bond. All right, so most of us picked the green one, so I think the person who I'm getting ready to call him knows that I'm going to ask them about the green one. Danielle Swice. All right, Danielle, why is the green one wrong?
Because So the attractive forces go with its nearby molecules.
Right, so it's gonna be like between a hydrogen and something else, right? Not between hydrogen. It's not gonna be forming between hydrogen atoms, right? But between a hydrogen and something else. And so this is where we need to put into that comprehension of also our choices at a level that we're able to understand it well enough. We got someone on here. The game code is, of course, I'm hiding it, I think. We'll see it on the next, oh, here it is. 547433. There it is, in case anybody else comes in later and they can see. Yeah, okay. All right, so exactly, that's exactly right. So it's gonna be between two different atoms. It's such a traction force. And that's an important thing to remember about our hydrogen bonds. Is we are not exchanging electrons like we are with covalent or with ionic bonds. With covalent and ionic bonds, we're actually involving movement of electrons, either sharing them or we have donated one to another atom, right? But with hydrogen bonds, it's meant to be attraction. And because it's a hydrogen bond, one of those atoms is always a hydrogen, and it usually has a slightly positive charge. It's going to be attracted to something that has a slightly negative charge. Usually that is oxygens, right? Oxygens are often electronegative and they hog the electrons. If you watch the lecture, then you hear me talk about how like two sisters that are supposed to be sharing Barbies, or if you're really mean like I am, then you steal the Barbie. That would be an ionic bond. If we're supposed to be sharing my Barbie with my sister, we can share them equally, like carbon does with hydrogen, shares the electrons equally. Or we can literally, like I am a mean sister and I hog the Barbie more. That's what oxygen does. Oxygen hogs the electrons more. Okay. All right, moving on. Next question. All right, what element is in all organic molecules? Should be an easy one for you guys. So I have a slightly different question on this one. I'm not necessarily going to ask the person to tell me why a wrong answer is wrong on this one. But do we have Catherine here today? Catherine Fields. Okay, Catherine. So what I'm going to ask you in relation to organic molecules, is it enough to only have carbon? Or do we need other things, something else to make it an organic molecule? So for example, is carbon dioxide an organic molecule? Why? It does. Do you know what? It's not enough to just have carbon. So does anybody know why? Yes. It needs hydrogen there. So we need a carbon hydrogen backbone. So I'll have you repeat that back to me. Why is carbon dioxide not organic? It needs hydrogen there. Exactly. Yeah, you guys will be tested on this, and it's not going to be definitions recall stuff. So we need to start getting comfortable talking about this. And it'll get easier and easier as I call on you more and more. But this part where we're thinking about this stuff and verbalizing it, it's different than just picking an answer on a screen or on a test, right? You have to think about it. You have to have some logic there when it comes out of our mouths. So it's literally me forcing you to do that. True or false? Water is a poor solvent. Water is this true or false? Water is a poor solvent, or I don't mean POUR. Is it, or is it a good solvent? Hopefully we know that water is a great solvent. It is the solvent of life, in fact, how all biological molecules, how life is actually suspended and how it works. So the question for this one is, why is water a good solvent? So this is for Sammy Adams. Sammy, are you here today? Sammy not here? Oh, she's on the Zoom. Okay, Sammy, you're here today. So why is water a good solvent? Can you tell me why? Yes, you are. You're all right. Take your time. What is it about water that makes it a good solvent, Sammy? Okay, so we're getting in the idea here It says you can delete and pair with things that you're working with. This isn't wrong, right? So we know that in water with H2O, the H can kind of come off and work with things, and the OH can come off and kind of work with things. And we kind of have constant dissociation between H2O and the H and the OH. We consider that to be just constantly in flux in water, right? But the thing that really makes water a great solvent, does anybody know what I'm getting at with this one? Anybody have any guesses? What do you think?
Is it because it's polar?
Because it's polar, because it can interact with other polar molecules. And in life, a lot of our molecules, because they have oxygens and things like that in them, they have charges to them, right? So it's really important to know why water is a great solvent. And this is extremely important when we start talking about the proteins later on. If you think that us bringing up this chemistry chapter, and we're not going to talk about chemistry again, I hate to break it to you, we're talking about chemistry almost every single day, constantly, because we need to understand these cells at the molecular level. It's really imperative that we start thinking about it that way. The way proteins fold is because we are a water-based organism and it's got polarity in it. So if we're 97% water, our proteins will interact with the water by putting their hydrophilic, the water-loving molecules on the outside, and the hydrophobic ones hiding them away on the inside whenever they're folding up. And this is key for how our proteins are going to function and things like that. So understanding how and why water works as a solvent is actually imperative. All right. What is water's pH? All right, good. I'm glad that we know this one. So this is a little one harder one to ask about, but I'm going to try to ask this one and see if we can get to it. Taylor Mala, is that how you say your last name?
Male.
Male. Okay, easy enough. All right, Taylor. Why don't you explain to me why it's pH 7? Like, give me a rough explanation of it.
Okay, well, I just think about it in the scale of like pH is 0 to 14,
Mhm.
7 is neutral right in the middle, and so water is like a neutral, you could say balanced.
Right. So that's fair enough. And so we're getting to the idea of it. If we break it down at the chemical level, if you have H2O and it splits up into H's and OHs, like we know about the H pluses and the OHs negatives, the hydroxyl groups, right? So whenever you split water like that, half of it is H pluses and half of it is OH negatives. And that would make it be right in the middle of the pH scale. That's why it's neutral, because we have equal amounts of H pluses and equal amounts of OHs to the H pluses. And that's why water is right in the middle of the pH. That's why it's neutral, okay? Because we know the pH scale depends on both the H pluses and the OH negatives, right? And then those interactions and their balances. If you have more H pluses, you're more acid, right? If you have less H pluses or more OH negatives, then you're at the alkaline side of things, right? Okay. So that's how our pH scale works, and that's why water is neutral. Pretty good answer there. We're moving on to our next question. Question 6, what range on the pH scale is an acid? Kind of just talked about this, but that's all right. All righty. We know that it's less than 7, right? Less than 7 is going to be our range on the pH scale. So Kaylee Jackson, are you here today? All right, Kaylee. So go ahead and tell me what this one would be, the green one. So we know acid is the red one. That was the right answer. So what would we call that? Basic, that's fine. Yes, basic or alkaline is another term we would use for that one, right? And 7 is neutral. Anybody know the pH? Sorry, you have a question? Okay. Anybody know the pH of the body off the top of their head? Yes. Close, yes, 7.35 to 7.45. So just slightly a little more alkaline than what water is, because all that stuff we have added in our blood, all the buffers and things like that. And so that's key. So when we say it should be 7.35 to 7.45, if you're deviating at all out of that, we're going to be concerned in the medical field about that, because it is actually really imperative that we keep it in that range, because more H pluses means that your proteins might not fold correctly. Yes.
This is kind of funny, but yesterday at work, we had a blood pH of 6.5.
What? Like, we're concerned if it's like 7.2.
Yeah.
What? So it's a guy that gotta be, was it like ketoacidosis?
It was a very, very like critical ICU patient,
Mhm.
but that one, it went on our record board at work.
Yeah, we see this most commonly, right, with people who have diabetes and they aren't managing their sugar levels well. And so then their body starts breaking down proteins, and then we start having acid production as a product of that. And then that's, some people will force themselves into ketoacidosis when they go on the keto diet, but it's balanced in a way, hopefully, with the other foods that you're eating, which is why we have a levels of how many carbs you should still be getting. If you're doing keto, you need to still be getting some carbs. You can't go without them entirely and stuff like that. So managing that can actually be pretty devastating. Does anybody know what body system is going to be affected the worst if that starts changing? Your kidneys. So your kidneys are the ones that are trying to maintain that balance in your blood. And so a lot of that work is done in your kidneys. And so if you are too acidotic for too long, then you will have kidney damage, and that can be permanent, in fact. So something to be aware of.
So do things like antacids, like Tums and stuff, like actually help?
It will. So the good thing about stuff like that, and I'm sure you guys have seen stuff about this on like Instagram or wherever, where they talk about like the alkaline water and stuff like that. So with Tums, it's a higher concentration of very basic alkaline tablets that's meant to reduce the acid level just in the stomach, and it shouldn't affect your bloodstream. It should only be localized within the stomach. I'm sure you guys have also seen, like, if you drink alkaline water and they're like, it's so good, it alkalizes your blood and whatever, blah, blah, blah. Your stomach acid will neutralize that within a second, and your blood will never see any benefit from that. And so if you feel better because you're drinking alkaline water, it's a placebo effect, and congratulations to you. Okay. And there's nothing wrong with a placebo effect, right? They're actually been proven by science to actually be helpful for some things where appropriate. But if you think you feel better, that's what my GI specialist said to me. I showed him these like digestive enzyme pills, and he's like, if you feel like you feel better when you take them, you just keep on taking them. And I'm like, okay, I get it. So, yeah. Anyways, great questions, guys. Good. All right, question 7, into lipids. What can you tell me about lipids? What are lipids for? All right, lipids. So this one's an all of the above. So I don't know how I want to approach this one. This one is going to be for Jimena. Are you here? Okay, yes. So this one, let's say, I don't want to get too tricky with this. So we have, it can be used to make energy for the cell. I don't know, this is the hard one. We'll come back to you. We'll do you for the next one. Okay, this is the hard one. It's a weird one to ask a question about. So anyways, yes, so lipids, we know that they make up the cell membrane. Actually, here's a question. What molecule with lipids is in the cell membrane? Do you remember what it's called? This is one that is amphipathic and should be one that we should have addressed in our little concept map if you made one. So it has a charged polar head and it has long hydrophobic water-fearing tails. Phospholipids, right? Okay. So phospholipids make up our membranes. We can use lipids to make our hormones. So our sterols are often used as a base for our hormones, whether that is testosterone or estrogen or things like that. They're often based on the sterols, which are lipid molecules. And one of the most important lipid molecules that can be part of our membrane is cholesterol. And so that is actually something that adds a little bit of rigidity and shape to our cells. The more cholesterol you have in your membranes, the more rigid your cell shapes will be. Obviously, that's when it's part of your membranes. Now, when cholesterol, because it's a lipid, is it going to mix well with our blood? No, it's a lipid, right? So most lipids are hydrophobic, right? And so is cholesterol. And so if it's not part of your membrane and it's, part of in your bloodstream free, it doesn't mix well with your blood. And so what it'll do is basically build up in your blood vessels and just stick to each other and not mix well with your blood, and then create those plaques that we see sometimes in our arteries and veins. Right. For energy for the cell, we know, and you've probably learned in chemistry, you should have learned in chemistry, that if you go through glycolysis, we're breaking down the sugar to make energy. But we can break down some of our fats and put those intermediates into like Krebs and things like that. So we can get energy from aerobic respiration, which you'll learn. Don't worry, we are not done with aerobic respiration. We're going to go back over it in chapter 10. It's not enough to just say you should have learned that in chemistry, because we're going to go over the details of it in chapter 10. Which of the following organic molecules is amphipathic? Oh, I just said it. Do you remember what it is? The phospholipids, right? Okay. So I'll give you credit for that one. So I will ask the next person about one of these other things a little bit. So we'll wait for you guys to answer. Next person. Next person's gonna be Jordan, by the way. Jordan Stallman, are you here today? Okay, awesome. All right, what can you tell me? What are amino acids for? You remember what we use them for?
I know they go from.
That's what I'm asking. So that's a general question. Yes, yes, yes. So we put amino acids together to make our protein. Nucleotides, what are they for? DNA and RNA, pretty easy, right? Phospholipids, those are going to make up our membrane, and they are amphipathic. Does anybody remember what amphipathic means? Well, we got both. We got both hydrophilic and hydrophobic on it, right? Amphi will always be both, just like amphibians. They can breathe outside of the water and they can live in the water. So I remember how amphi is at the same time, right? Glucose falls into our carbohydrate category. Does anybody know, is glucose going to be polar or non-polar? Anybody have a guess? So what do you think, polar? We got votes for polar. Yeah, it's going to be polar. And one way we know that is how many oxygens are freaking in one of those rings. Remember, if you learned about it in chemistry, how you have oxygens popped here or there, OH groups coming off of there. Those oxygens being very electronegative. They're always electronegative. They never change that. And so if it's got oxygens, it's almost always going to be polar. And so it's almost always going to mix well with water. One way you can know too is think about in real life applications of sugar. Does it mix pretty well with water? It's pretty easy to mix it in because it's polar. So if it mixes well with water, it's probably polar. Okay, so good thinking, guys. Excellent. That was Jordan. We're doing great so far, and you're keeping up. Next question. What is a monomer of a nucleic acid? We just mentioned it. Let's see if we can remember it. Nucleic acids are made up of these parts. So this is going to be the nucleotide. So a monomer of a nucleic acid. So nucleic acids like DNA and RNA, they are polymers. And so their subunit parts are called monomers. And so those things that make up the nucleic acid, that's why the answer is nucleotide. Now, DNA is a type of nucleic acid, but that's not the monomer of it, right? Okay. So the thing that makes up DNA is still nucleotides. So that's why nucleotides the answer and DNA isn't. Well, I'm going to call on somebody for another part of this. Lauren, are you here today? Okay, Lauren. Pentose, what does that mean? Don't read too hard into it. Don't read too. So when we think of the pentagon or.
Yeah, it sounds like, you know, the glucose.
Yes.
And,
the oses. Okay, so the oses does signify to us a sugar. And what does penta mean, number-wise? 5. There we go. So what do we think pentoses are? 5 carbon sugars. Excellent. So important for our nucleic acids. Ribose and deoxyribose are pentoses. So ribose is the base that went in there, right? It's a 5 carbon sugar, ribose. We know that that's an RNA. Deoxyribose, what do you think the difference is? And oxygen's removed. That's it. Deoxide. We removed an oxygen. So I will never make you draw it or recognize a picture of it in my class. Okay. But knowing that is a little helpful in understanding the difference in the structure. Okay, but ribose, 5 carbon sugar. So therefore it is a pentose. Excellent. We worked through that pretty well. Good job, Lauren. All right. It's also a name I know how to say because it's my name. It makes that one easy. All righty. Question 10 for chapter 2. Alpha helices, or helices would be better, and beta pleated sheets. Which type of protein structure is this? Where do we see alpha helices and beta pleated sheets? Which level of protein structure? Excellent. So this is going to be the secondary. Most of us got this one. Definitely, this is going to be because of the neighboring R groups on our amino acids. Okay, so those, if they're hydrophobic or hydrophilic or polar, if they're attracted to each other, or if they repel one another, just makes those initial shapes. It's like whenever you have a ribbon and you've curled the ribbon before you tie it into a bow. Okay, so this is the secondary structure, and it's because of the neighboring R groups on our amino acids that are linked together, making our polypeptide. All right, Kay Sanders, are you here today? Kay, okay. Can you tell me what the primary structure is? So like the blueprint of the protein, and quite literally it is made up of just the amino acid sequence. So when we learn about those 20 different amino acids, alanine, phenylalanine, glutamates, leucine, whatever else, other ones, there's 20 of them, and you don't have to memorize them or know their structures. But there are 20 different amino acids. Basically how we would spell out the protein, we have methionine, isoleucine, glutamate, whatever, however you spell out your protein with your amino acid, that's the primary sequence. So that builds your ribbon. And when you have the immediate groups next to each other and how they behave to each other, that makes our alpha helices, which are helixes, or beta pleated sheets, which are like straightaways. And they'll like fold back on each other and be just straightaways like that. That's our secondary structure. Our tertiary structure is how that overall structure is going to behave with one another. This is where we'll start having those disulfide bonds across the way. And so this is where we start holding to have our hydrophobic on the inside and the hydrophilic on the outside. Overall big major structure of the polypeptide. Yes.
Wait, so is this the one with the AT or AUG?
That is your DNA and your RNA. So DNA is ATCG and your RNA is AUCG. So we'll learn about that in chapter 6. It's okay if you guys don't quite remember that stuff. Chapter 6, that's a unit 2 thing. So don't stress too much about that just yet. Chapter 6 will cover that stuff, okay? But your DNA, that's your genetic code. And then if you have AUG, that would be a codon. That would code for methionine, and methionine would be the one amino acid that comes to you. We're calling this a little bit. Yes, that's translation, right? So that's chapter 6. We'll review that stuff. So you don't have to have like your basics down if you lost some of that. We're starting to think in that direction a little bit. Yes. Okay. Great. Trying to think what else I want to say about this. Good. Okay. Cool. All right, let's look at our podium. See how you guys did. All right, and I didn't get to quaternary. So think of like with our protein structure, you got your amino acid sequence. You've got a couple curls where you curled your ribbon. That's your secondary from the R groups that are next to each other. Tertiary is when you tie that one ribbon, but whenever you're wrapping so much present, you might want a big fancier bow. When you want a big fancier bow, you'll have multiple bows that you'll put together. That's the quaternary structure. Is when you have the whole big package bowed up with multiple ribbons. That's the quaternary. Okay, anybody have any questions about that stuff? I'm going to pull up the next one so that we don't forget about it. Chapter, what is it next, 4? Okay, we'll come back to this in a second. All right, so next thing is going to be our activity for chapter 2. First question, and like I said, if they're kind of easier basic questions, then I might call on you guys. If they're more meta, complex thinking, then I'm going to hope that we'll talk about it as a group, and I want to call on an individual. All right, so our first question here, learning about our chemistry of life, how are electrons distributed in a molecular bond between a highly electronegative atom, like oxygen, and a hydrogen atom that is covalently bonded to it? So I'm going to call on somebody for this one. Jeremy, are you here today? Pulling you out of the ether. All right, Jeremy, what do you think? How are electrons going to be distributed between oxygen and hydrogen? Is it going to be equally shared? Is it going to donate it? Is it going to hog? One is going to hog or the other? Okay, so we can sometimes see that happening where we'll actually donate the electron from hydrogen. Because remember, hydrogen only has one electron. On the periodic table of elements, it's number 1. It's got one proton and therefore has how many electrons? 1 equal number. Okay? So it can actually donate its electron to the oxygen, but typically if they're covalently bonded, are we gonna see that? No, they're gonna be sharing. And so it'll be an unequal sharing. Because oxygen is electronegative, oxygen's gonna hog that electron more. So it just spends more time with the Barbie, like we were saying with my sister's example, right? So it's gonna spend more time with the electron. That oxygen hogs that electron. That's because it's electronegative, just has a tendency to want to spend more time with it.
Is that for hydrogen bonds?
So this is setting up for the hydrogen bonds, right? So because that oxygen now has a negative charge, what charge does the hydrogen have? It spends less time with the electron. It's like what we call the positive dipole moment, but we don't get that fancy in my class. But it's got a little bit of a positive charge, right? So now that hydrogen, so if we have a molecule that has an OH group, here's a molecule, I don't know, whatever, and here's an OH group. That oxygen, it's going to hog electrons, making it what charge? If it has the oxygen, if it has the electrons more often, it'll have negative charge, right? And the hydrogen will have what kind of charge? Positive. They'll still be covalently bonded. They'll still be sharing that electron between each other, but it's just going to spend more time with the oxygen, right? So this gives what in chemistry you guys would have drawn it like this. This little D thing means they have a dipole moment. And this is polarity. And so this is why the H plus is going to have a tendency to be a little bit more positively charged. Like true donation of the electron. Sometimes we can actually see true donation of the electron because it'll get so greedy, it'll sometimes steal it. Does happen. Most of the time if we're covalently bonded, so we're still sharing, just very unequal sharing. And so now we've set this up that this hydrogen, because it's positively charged, can be attracted to something that is negatively charged. Maybe let's say water. What's the oxygen charge in water? Right, we see this, right, in water. So now this is the actual hydrogen bond. It's an attraction between two different molecules because a positively charged hydrogen was attracted to something negative. Doesn't matter really what at that point, but that's the hydrogen bond. We tracking? Any questions about that? Okay. What is happening to the hydrogen concentration in a solution if the pH goes down? If the pH is going down, what are the hydrogens doing? Jacqueline? Yes. Yes. pH is going down. So do you know what we call that in our pH scale? Let's say it's less than 7. Anybody know? We want to help her out? So that'd be acid, right? So what's happening to hydrogen ions in an acid? We have more or less hydrogen ions? More. It's more. This is important that we get this one, okay? So with this, I'm gonna make this go up for a little bit because my pen on this doesn't work. So you're gonna, actually, let me try to type it out because I feel like the people that are online might wanna see this. Let's see if I can make it work. Annotate, here we go. Let's type it. Okay. So you guys can still see it. All right, this is hydrogen concentration. So pH, you guys hate this equation, I know you do, but you've got to remember what the actual equation is. Negative log of the concentration of the hydrogen ion. This matters. I know it seems like nonsense and it's just like, forget it, I don't care. That's how my brain feels about it too. But you need to understand what's happening here. Negative means the negative part of this inverse. That's the key probably to all of our pH issues. If I'm calling y'all out on those of you that don't understand about pH or you get confused on it. Negative means inverse. What does log mean? Does anybody know what that actually means? Logarithmic, what does that mean? 10 to the power of whatever you're talking about, okay? So if we say log of something, we're talking about at base 10, the base is 10. So if we're talking about something, if we're talking about the log of 7 equals 10 to the, this will be 10 to the 7, right? This will be 10 to the 7, okay? But if it's negative 7, it'll be 10 to the negative 7, okay? So if we're talking about the negative of something, that's the inverse. So if 7 is 10 to the 7, then negative 7 we could say log equals, actually say pH, we'll just leave it at pH. pH equals the negative log of our concentration of our hydrogen ions. Okay, so let's say it's 0.01. What's 0.01 in exponents? Anybody know? 10 to the negative 2, right? 10 to the negative 2. This part's key. What pH is that? What pH is 10? What is that pH? 2. That's where the number's coming from. So we have a pH of 2. We're talking about the concentration of the hydrogen ions being 0.01. If we have a pH of 10, try to write it out when we put it up here because we have some space. Okay, guys, so you can actually see what I'm doing up here. So pH of 10, what is our hydrogen ion concentration? It's going to be 0.1 would be 1, pH of 1. 0.01 would be pH of 2. 0.001 would be pH of 3. 0.0001 is 4. So we would go take this all the way out to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Which one's a bigger number, 0.01 or that? 0.01 is a bigger number than this, right? And so this is the literal concentration of the hydrogen ions. So when you have a pH of 10, that's your concentration of your hydrogen ions. When you have a pH of 2, it's 0.01. So now you can actually see that the concentration of the hydrogen ions is more with pH of 2 because we're actually talking about 0.01. Are we tracking? And so when we get off of a pH of 7, right, what we have with water, your hydrogen ion concentration, it has the 7 numbers after the digit, okay? But your, so is your OH. They're equal in number, okay? And anything more than that, we will have acid. Anything less than that, we will have base. But when we talk about pH, don't focus on just knowing your pH scale numbers. Know what's actually going on with our hydrogen concentration, because I will ask you questions about that on the test. I will tell you, if your patient's urine, normal urine pH is supposed to be 8, and your patient's urine pH is 9.5. Does it have more or less hydrogen ions than it should? It has less, exactly. So that's the type of thinking that I do expect on my test. I'm not going to give you the pH scale, and I'm not going to tell you what pH means. I'm not going to tell you what it represents either. You will have questions like that on the test. Okay? I expect that you guys would have learned this in chemistry. I know it's been a while, right? It's been a while, and y'all all thought that you would never use it again. But I'm already telling you, we're not maybe going to use, this is one time when we have an equation we need to be aware of. One time. Promise, this is the only one, okay? And I'm not going to make you do calculations with it. I just want you to be aware of what it actually represents. When we say pH of 2, we're talking about 10 to the negative 2 is your concentration. If I say pH of 10, 10 to the negative 10. And now break it down as our decimal. If you need to break it down as the decimal to compare it, do it. Right? If you need that. Okay, we're tracking? All right, 4 major groups of biological molecules. We could call on somebody for this one. This is not hard. Kaya Hughes. Here. Okay, can you name one of them? So there's 4. Definitely should track this in our concept maps, right? Right? Yes, we can't forget it after that though, right? Because y'all gonna get tested on this stuff, right? And that's what we're here for today. All right, who wants to help her? Name one. Carbohydrates, anybody else? Lipids, okay. Carbohydrates, lipids, nucleic acids, and proteins, exactly. So we'll come back to you, Kai. We'll try to see if we can get you an easier question a little bit later, okay? All right, great job. So we got that, carbohydrates, lipids, nucleic acids, and proteins. Give one example of a hydrophobic molecule in biology that was discussed in this chapter. I talked about a few earlier on, and Kai, if at any time you know the answer to one of these, just raise your hand and let me know, and we can get you credit for that question, okay? So anybody know an example of a hydrophobic molecule that we talked about. So I don't want you telling me phospholipids, because phospholipids aren't technically hydrophobic, are they? What are phospholipids? Amphipathic, okay? Anybody know a hydrophobic? Yes. So benzene, yes. Benzene is hydrophobic for sure, but is it one of our biological molecules? Technically, it's not. And so we'll see benzene rings as part of the molecule sometimes. But overall, yes. So in this chapter, one of the, the core chemistry side of it, yes. Here, I'll give you guys an example, cholesterol, which I talked about earlier. Cholesterol, hydrophobic, all right? We also have our fatty acids are tricky. The tails on the fatty acids are definitely hydrophobic. But at the top of it, we have a glycerol molecule. And glycerol has a lot of oxygens in it. It's fatty acid, right? That acid part of it gives it an amphipathic quality. A little bit. Is it largely hydrophobic? It's largely hydrophobic, but it's a little bit amphipathic. But cholesterol is one that we can say is generally largely hydrophobic. Given what we have learned about proteins and their structures in this chapter, why might a decrease in pH negatively impact a protein's ability to function? All right, so think about that. If we talk about our R groups on our amino acids, we got a lot. Glutamate, for example, is a charged one, right? Yes, what are you going to say?
Is it because the protein structure is cut together with hydrogen bonds?
Right. Right, exactly. So a lot of our folding, even our alpha helices and beta sheets, those interactions depend a lot on hydrogen bonds. If we're depending on this attraction, our hydrogen to our whatever oxygens typically is what we're attracted to, okay? If we're depending on that interaction to create some attraction and hold our overall structure of our protein together, if you get a lot of H pluses out here that are extra, which is what would happen if your pH goes down. pH goes down, then your hydrogen concentration goes up, right? So you have a whole lot of hydrogens out here, and they're going to interact with that oxygen, and the other molecule might not. And so you can lose the structure of your protein pretty quickly because you're disrupting those hydrogen bonds that hold your protein together. And they might not disrupt your cysteine disulfide bonds that we learn about, right? They might not disrupt that, but a lot of your structure depends on hydrogen bonding and polarity and interaction of molecules that are charged interacting with each other nearby. So once you have too many H pluses or too few H pluses, those interactions don't happen the way that they're supposed to. So it disrupts how your protein is folded. We call that denaturing the protein. And you start unfolding it that way. And that's a permanent process. Just like when you cook eggs, right? Can you turn that egg back into a raw egg? No. Once you've denatured those proteins in that egg, it's denatured. Okay, so we can actually use pH as an ability to control microbial load and things like that whenever we are cleaning surfaces and stuff, which is why vinegar can be used in household cleaning to control microbes. It'll disrupt their protein function. Because vinegar is an acid, if you didn't know that. The acetic acid is what primarily is in vinegar. In fact, if you smell glacial acetic acid, which is pure acetic acid, it is like the strongest vinegar smell that you've ever smelled. It's just so strong. All right, anybody have any questions about these questions? All right, because moving on, starting with chapter 4, which we're going to do here in a moment, moving on from chapter 4, a lot of them are going to be actual case study based from like an actual scientific article. So we want to be kind of aware of this is our intro into it. But next we're going to be assessing scientific articles, all right? Just to show you guys, this is what we'll be looking, maybe after I close this. This is what we'll be looking at here, right? Nope, just by being here in class, you guys get credit for it. My online class has to post them, but you guys don't. Just by being here and participating, you get credit for it, all right? But the idea is that if we're walking through this in class, that you guys are tracking why I'm answering the question the way that I am. And we're engaging and discussing why those answers are right and stuff. Make sense? All right, we're gonna come back to our Kahoot. You might need to sign back in. Let me make sure this is. Why? I can't see the bottom of this. It's not letting me. Whatever. Here we go. Okay, make sure you sign in. Give you guys a minute to sign in. We've got exactly 10, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We've got 11 questions to ask you guys to make sure everybody's been called on. Y'all know you're coming if I haven't called on you yet. Right, make sure everybody signs in. Are we ready? Go ahead and get going. All right, first question for chapter 4. This is our bacteria and our archaea. Mostly bacteria, if we're being honest. Since bacteria don't have a nucleus, they can't contain DNA. Is that true or false? Righty, so this is false. So I'm going to call on Maddie. Maddie, are you here? Okay, awesome. All right. Explain to me why this is false.
Because bacteria, they don't have a set membrane-bound nucleus.
Mhm.
They have what's called, I think, a linear or circular form of DNA.
Yes.
It's basically instead of the X's for chromosomes like we have, they have one long strand of DNA.
Yep, they do, and they still have DNA. It's just in one big old circle, one continuous circle. And that's where all of their genes are. Are they going to have less genes than us? Yeah, quite a lot less. But they'll still be fully able to replicate themselves. They're still cells, but they just don't have a membrane-bound nucleus. They still have DNA. We're going to deviate from the DNA being the genome when we get to viruses. Viruses are the only ones we're going to see that might not have DNA as their genome. What else could they have as their genome? You want to guess what the other thing is that they could have as their genome? Still got to have a genome. It's not DNA. What do you think it's going to be? RNA. So they can have a lot of different versions of that too. Because we know our DNA, if you're a cell, you have double-stranded DNA. Double-stranded in a double helix, like we know the ladder that's all spun up and whatever. With bacteria, it's one continuous circle. With eukaryotes, it's in linear lines, like we think of the X's. Okay, we have several of them. But viruses, they might have multiple strands, they might have single strands, and they can have double-stranded DNA, they can have double-stranded RNA, which is wild. We don't have that in our cells at all. No double-stranded RNA in our cells at all. In fact, if your body sees double-stranded RNA, it will immediately say, bad, foreign, out, out, out. It's one of our ways that our body can immediately notify us that something is foreign. We'll talk about that in Unit 3. Unit 3 talks a lot about how our bodies kind of can tell the difference between something being bad guy versus self. But that's an indicator right away. Our cells don't have double-stranded RNA. So if our body sees double-stranded RNA, that's one way it knows. Another thing that our body doesn't have is peptidoglycan. You might have bacteria in your body, but your actual cells, do they have peptidoglycan? We should have learned that in chapter 4. But that's something that we see in bacteria. Where in the bacteria do we see peptidoglycan? What part of the bacteria is the peptidoglycan found? The wall. Yep, the gram positives and the gram negatives. And if you're sitting in this class and you're like, yeah, I don't know that, you are in a bad place. Because I mean, that is the bread and butter of everything that we're going to be talking about moving forward. I'm going to be saying gram positive and I'm going to say gram negative. Chapter 4 is where you learn that, and you need to carry it with you because I'm gonna expect you guys to know it, okay? Because it is the key to it all. Will I bring it up and explain it over and over again? Yeah, I will. Okay, so you won't get lost in that. All right, moving on. Next question. I'll remind you constantly about it, but definitely try to hold on to that one, right? Peptidoglycan. All right, here we go. The peptidoglycan layer is blank in gram positive compared to gram negative. What's the difference? What's the difference between gram positive and gram negative? That's really what this question is asking. All right, so it's substantially thicker. In fact, in gram negative bacteria, they have, almost always have, some peptidoglycan, but it's usually like one layer thick. So what do I mean by a layer? With peptidoglycan, Get the pen. Hold on. I don't know. Whatever. All right, with peptidoglycan, we got sugars. You guys know how sugars are drawn, right? We got that hexose thing going on here. It's a terrible drawing of it, but you guys get it. All right, we got our hexose thing. These are definitely the same kind of molecules, even though they look super janky. And these hexoses are linked together in a nice long chain. And then we got another chain of hexoses down here. If I could copy it and just paste it, it'll probably be easier, but we're going to draw them and they're getting bigger and bigger for some reason as I draw them. All right, so we got like a whole chain, and then this isn't just two next to each other. We got a whole bunch next to each other. And these are two different kinds of sugars, and they're alternating. One is NAG, which I'll put as G, and one is NAM, which I'll put as M. And you don't need to know that. I'm not going to test you on that, like pretty much every other microbiology class will. But I just don't care that much that you know that it's NAG and NAM, which is N-acetyl-muramic acid and N-acetyl-glutamic acid. They're sugars. That's what I want you to know, okay? So we got these layers of sugars. If you guys can look at the word peptidoglycan, glycan is the part that says sugar to us, okay? Like glycogen or glucose or something like that. That glycan part is the sugar part. And these are linked together with little cross bridges. These are little peptide bridges. So what's a peptide? Does anybody know? What does that mean? It's a small chain of amino acids. That's what a peptide is. Usually 20 or less, we call it a peptide. Okay. So polypeptide is when you get over 20. So our little peptide bridge is just some amino acids, isoleucine and alanine and all that crap that we talked about earlier. It's a little bridge of that connecting these chains. So peptide, amino acid bridge. Glycan, sugars together. And so this creates a big old scaffolding. And so it'll look like this, be like multiple layers. In gram positives, I mean, we're talking like 30 to 40 layers thick. And these are all like kind of cross bridge linked together. And you get the idea, right? We've created a big old scaffolding. That's the cell wall made of peptidoglycan. For gram positives, this is very, very thick. For gram negatives, this is very, very thin. All right, so we're getting to the part where I'm going to ask a question. I'm going to close this. Actually, let me bring it back up so I can remove it. I'm getting rid of my drawing just so you know. All right. Oh, Kate Henderson. We here today? Kate here? No Kate. All right. I know Brandy isn't here. I'm assuming Brandy isn't here. Brandy McAfee. She hasn't even logged in online, so I want to be sure that she's not here today. And so, of course, Saber isn't here either, right? Saber's not here. But also somebody that hasn't even logged in yet online. I need to do automatic withdrawals for the students. All right, Amna. So you come in late. There you are. Okay. Can you tell me what other difference that gram negatives have? So we only have a thin wall. There's one other key characteristic of gram negatives.
Can I tell you about this, how it stains differently, maybe?
Why not? Sure.
They stain pink.
They do. So we'll talk about that in the lab. We're going to do gram stains on Wednesday, right? So I'll go over a lot of like the staining part of that on Wednesday, but it does talk about it in chapter 4, doesn't it? So the whole reason that gram stains work is we built that scaffolding. The gram positives trap crystal violet, our primary stain, in the scaffolding. The gram negatives don't have anything to trap it with, really. It's very thin. So when we rinse out, we try to link in any crystal violet that gets stuck. We use a mordant. Mordant is something that causes things to stick to. Kind of like brick mortar causes the bricks to stick together. Mordants cause our stains to stick in place. And then our staining procedure of crystal violet, and the next thing we follow it up with for a gram stain is iodine. Iodine is our mordant that causes what crystal violet that got stuck in there to stay in place. It fixes it in place. And then we rinse it with a decolorizer. Anybody know what the decolorizer is? Ethyl alcohol, exactly. So it decolorizes any crystal violet that didn't stay there. When we go through this, I walk you step by step and show you what it looks like and how you can tell what's happening and that's going the way that it's supposed to. Okay. And then we counter stain with safranin, which is a red stain. It looks pink in the microscope, but I always say the red versus purple because purple positive, PP, purple positive, and red is a negative color to me in my brain. Maybe it is to you. And so I think of red as being the negative and purple positive.
Did they name that dye after saffron?
I think so, because saffron actually is a reddish color to it, naturally. Like when you look at the actual like stamens or whatever that they pick for it, I think they're actually red. But it dyes clothes yellow. It's like interesting. Yeah. Anyways, whatever. For what it's worth. Yeah, so I think it's named from that. Anyway, so the other characteristic that I was hinting at, so that's good. You covered the stain part of it. The other characteristic about gram negatives, they have a thin wall, but they also have an outer membrane. So that's what I was getting at with that, right? So gram positives, thick wall, but that's all they got. You give them a drug that breaks down that thick wall, they're screwed because they needed that thick wall to live, right? Gram negatives, they got a tiny little thin wall. You give them a drug that breaks down the wall, they don't really care as much because they got a whole other membrane on the outside. Give them support. So they've got two membranes, the inner cytoplasmic membrane and then the outer membrane. It's actually a key identifier with our gram negatives. And it actually has another molecule that's very, very significant in our gram negatives in that outer membrane that we call lipopolysaccharide. Yes.
Do gram-positive bacteria, do they have that cytoplasmic membrane
Yes.
too?
All bacteria will have the cytoplasmic membrane, which is our inner membrane.
Okay, so
Yeah.
the positive ones, they just don't have that.
They just don't have the outer, they just have the one membrane. So all cells, in fact, have a cytoplasmic membrane. What keeps with the goo in there, the cytoplasm is the goo. So the cytoplasmic membrane keeps the goo in. The gram positives have another wall outside of that. The gram negatives have a tiny wall and then an outer membrane outside of that. So key for the gram negatives is that outer membrane. They care more about that. All right, let's move on. Yes, okay, next. Cool. All right, the gram negative outer membrane. It has something in it. I just said what it was, but let's see if you can. It's not the word that I use. There's other words for it. Let's see if you know the word. Let's see if you know the other word. Gram negatives have a special molecule. I called it lipopolysaccharide, but we also call it something else. The lipid part of it, we call it something else. This is absolutely key in medicine. I'm telling you, with infection, this is important. This is key because in medical practice, we call it endotoxin. We don't call it lipopolysaccharide or the lipid A part of lipopolysaccharide. In my class, you'll hear me almost always, if I'm talking about lipopolysaccharides, if I didn't bring it up earlier, because I have a feeling there's a question about it coming up, because I don't have these memorized, right? I had a feeling it was coming up. Endotoxin is what we call it. And endo means inside, right? Right? Endo. But it's a toxin to us. It causes damage to our tissues and our cells. The bigger key is if you have a gram negative infection, your body immediately sees the endotoxin, which is the lipopolysaccharide on that outer membrane, as foreign, and it has a shock response to it. And this will lead to essentially toxic shock response. And it can be devastating to your blood pressure and things like that. We have a gram negative infection, especially when it gets into the bloodstream, and the bloodstream goes all over your body, and then you're having a shock response basically throughout your whole body. Even with localized infections, this can lead to severe inflammation pretty easily. So endotoxin of any gram negative bacteria. Some examples of gram negative bacteria, before you start thinking like they must be super rare, get that out of your head. Every single bacteria in your gut right now, almost all of them, except for maybe a random few, is gram negative. So why don't we have our inflammatory? I'm telling you, it immediately triggers inflammation. Your body had to train your immune system that when there's gram negatives in your gut, we don't freak out from that. And so this is why this is a pretty serious issue. When you have GI illnesses, they're often caused by gram negatives because your body kind of sees it as it's okay for gram negatives to be there. But salmonella doesn't belong in your gut. Does it go there and grow? Yeah, if you get infected with it, right? But your body doesn't see it as immediately responds to it because we've trained our gut to think gram negatives belong there. So we have to wait for your body to catch up with this being foreign. We have to find something else foreign about it. It can't just be that it's gram negative. But everywhere else in your body, we shouldn't have gram negatives in a lot of other places in our body, like on your skin. We shouldn't have skin infections. We shouldn't have your CNS. Your like cerebral spinal fluid, your central nervous system shouldn't have any bacteria in it. So if we see endotoxin, LPS, in those areas, it will trigger pretty severe inflammatory response. The worst possible infection that you can get with meningitis is caused by gram-negative bacteria. So this is Neisseria meningitidis. It's the one that causes the rash. Probably heard of this. Rash is petechiae. Anybody know what petechiae are? What is petechiae? It's little ruptured capillaries. You've probably heard of a petechial rash, like a spotted little rash, but those spots are actually ruptured capillaries. So what happens is, whenever you have meningococcal meningitis, which is Neisseria meningitidis, that it causes your body to respond to the endotoxin, and it bursts your capillaries from all the inflammation that your body's having to this bacteria because of the endotoxin. So that's the second most common bacterial meningitis. The first most common is caused by Staphylococcus pneumoniae, which is a gram-positive. So you can see already, the most common bacterial meningitis is caused by Streptococcus pneumoniae, which is gram-positive. Much easier to treat as well, and much less deadly. It's the pneumococcus. Okay, so knowing the difference between the pneumococcus and the meningococcus, just knowing their shape, you can already tell if you've learned in chapter 4 about the cell shapes. The coccus, what shape is coccus?
Circle.
Circle, round. They're both cocci. But if I take your cerebrospinal fluid and do a gram stain on it, I'll see that they're cocci, but I'll also see that they're gram-positive or gram-negative. If it's gram-positive, much better prognosis for your patient. If it's gram-negative, we've got to work because you will most likely die from that if you have it. It's very serious. In fact, if you come to a hospital with meningitis, we're going to immediately treat you as though you have that worst kind of meningitis. We don't care yet. We don't know if it's bacterial or fungal or viral yet, but we will find out. And for now, we're going to push all of our heavy antibiotics onto you and cross our fingers. And yes,
Whenever we have meningitis, people come in through the ER, they're like, we put them in the airtight room.
Yep.
Full PPE, everything. It's
a very
crazy.
do not poo-poo on a meningitis. I'm telling you, when you guys get into nursing school, they will drill this into you so hard. If your patient has light sensitivity and neck stiffness and a headache and a fever, that's meningitis, and we're going to treat them like they have the worst meningitis you could possibly have, which is the meningococcal one. Until we know better, we treat them that way. Once we know that it's viral, we'll be like, oh, thank God, they'll get over it. A lot of people think viral infections are worse than bacterial. It depends on the illness. Okay? Any day, I will take a viral meningitis over a bacterial one. Any day. Because most people recover easily from viral meningitis. But bacterial meningitis, once you're actually having symptoms like that, you're probably in a really bad way. And if it's Neisseria meningitidis, meningococcus, then that can become septic very easily. And that leads often to limb loss and things like that very quickly, actually, because of the endotoxin and the response our body has to it. Okay. We're tracking a little bit on that and seeing like why we're even learning this. I hope that we're seeing why we're learning it. All right, moving on. Question 4. What is acid-fast staining? Acid-fast. Why do we do it? What does it tell us? And I am going to ask a question. Is London here? London with a Y. Okay, London with a Y. We have 2 Londons. So London McNair. I'll call you by your last name, not just London with a Y. All righty. So acid-fast staining is staining to determine the presence of mycolic acid. Okay. We'll learn acid-fast staining. It's lab number 4. So we'll get into the details of it when we get to lab 4. But I am going to ask you, London, what is mycolic acid? General. Part of the cell wall. Okay, so that's good enough for me. We're getting into this thinking. Mycolic acid is a special wax that is found on the surface of some gram-positives. So very few gram-positives. There's only 2 genera of gram-positives that can be acid-fast. So acid-fast determines how they're going to stain. And what it means is we're going to stain them with a special stain called carbofuscin. And carbofuscin is a lipid-soluble stain. That's the important part. Okay? Lipid-soluble, which means it stains things like waxes. You know, waxes are hydrophobic. I hope we at least know that, right? When you mix your candle with water, does your candle wax mix with the water? Right, it's hydrophobic. So it's a wax, all right, mycolic acid is. And we use a special stain that is lipid soluble to stain just the wax. And then when we rinse the stain out, just like we did in our Graham stain, we rinse with the ethanol. But with the acid fast stain, we rinse with a little bit of acid. And what it does is if you are a normal cell that doesn't have wax on it, you won't hold fast in the presence of acid to the lipid stain. You won't hold fast. But if you are acid fast, you hold fast to the stain in the presence of the acid. And that's what it means. Like you're holding, like I always think of like on a ship, you're holding fast. Hold fast while you're like on like the seas or something like that. That's what I think of when I think of fast here. Fast does not mean speed. It's like holding fast. Okay. And the cell will hold fast to the stain if it has this wax, mycolic acid. So they are acid fast. All right. That's what that means. So acid fast means you have this wax, mycolic acid. Only 2 genera have it. So genera is what our first part of our name is in our binomial nomenclature. We remember that, right? So that's from chapter 1. So Escherichia coli. Escherichia was the genera. Are there other Escherichia besides Escherichia coli? Oh yeah, there's plenty. Let me tell you about Staphylococcus and Streptococcus because it goes wild. All right. But the 2 genera that can be acid fast are Mycobacterium, not Mycoplasma, Mycobacterium, and Nocardia. And I will never test you on Nocardia because it's not medically relevant. I will test you on Mycobacterium. Anybody know one disease caused by Mycobacterium? Yes. Tuberculosis. And you know another one? Anybody?
Leprosy.
Yes. Leprosy. Those are 2 big. Leprosy is being very close to almost eliminated from the planet due to treatment with antibiotics. It's a very slow growing bacteria. It only doubles once like every 45 days. It's wild. So you only go from 1 cell to 2 in 45 days. And so culturing it is very, very difficult. So we usually can't culture it. Mycobacterium tuberculosis, on the other hand, because tuberculosis, it's a little less slow. Still kind of slow, but it's a little less slow. It's a BSL-3 pathogen, so it's one that we would put in a containment room. And it's a little bit dangerous to work with there. About 1/3 of the world's population has tuberculosis. Good question, Maddie.
What's the difference between a mycoplasmic bacteria and then a myco?
So we have to know that difference too. I'll tell you about the difference really quickly. Mycobacterium. When you hear mycobacterium, I want you to think about bacteria with myco, okay? That's what I want you to hear when you hear mycobacterium. And what's the myco? Mycolic acid, okay? When you hear mycoplasma, I want you to hear plasma. Forget the myco. Mycoplasmas do not have mycolic acid. They don't. Mycobacterium do. Anybody know any relevant mycoplasmas off the top of their head? May have heard of it, may not have. Mycoplasma pneumoniae. So you might immediately be like, oh, probably causes pneumonia, and be right.
It's the walking.
It's walking pneumonia. Walking pneumonia. So if you have pneumonia, fluid in your lungs is what pneumonia is, okay? So you have pneumonia, but you're able to function and go to work or whatever. Otherwise, you still might feel like crap, don't get me wrong. But you can otherwise function. You're not hospitalized and on like a ventilator. Then you have walking pneumonia. If you are so cyanotic blue, you aren't getting enough oxygen that you can't function normally day to day, that's actual pneumonia, like true, like what we consider typical pneumonia. But walking pneumonia, also known as atypical pneumonia, you're able to continue functioning in life. This is almost always caused by Mycoplasma pneumoniae. What's the significance of, well, let's talk about the bacteria side of the mycoplasma. That's why we're even talking about this. We know Mycobacterium, it's always in the name. Mycobacterium will be the name of whatever it is. Then it has mycolic acid. Mycoplasma, again, in the name, focus on the plasma, and this is why. It's how I remember the difference, okay? If you're carrying a cup of plasma, like literally somebody donated plasma at the blood bank, and you have a cup of plasma, I don't know why you're carrying a cup of plasma, but you are, and you trip and you spill the plasma, does it hold its shape? No, it doesn't stay in cup shape, right? It comes out and spills everywhere, it's liquid, right? So if you spill a cup of plasma, it doesn't hold its shape. Mycoplasmas, they don't have a shape. They're pleomorphic is the word for that. They don't have a defined shape. Why don't they have a defined shape? Because they lack a cell wall. They don't have any peptidoglycan. Yes.
Why does it have, but why does it have myco in it then?
Don't worry about it. Don't read too much into that. But that's why I say to you guys, if I talk to you about mycobacterium, this is because obviously people mix these up, right? If I say mycobacterium, bacteria, focus on that first. Okay, it's bacteria. Does that tell you anything special? No. Then come to the myco, and it tells you it's bacteria with mycolic acid. If you see mycoplasma, oh, that was the cup of plasma that doesn't have the shape, then focus on that.
So mycobacteria is what the acid fast is testing. Okay.
Yes. So acid fast test for mycobacterium species. Only mycobacterium species are acid fast. They're the only one. Yes, no cardia, but like I said, I'm not going to test you on it, so just move on. Just move on. We don't need to be myocardia. Cool. Mycobacterium are the ones we care about. They're medically relevant. A third of the world has tuberculosis. That's medically relevant, right? All right. Are we tracking? We're getting there. All right. True or false? Mycoplasma is a acid-fast positive organism with a thick waxy cell wall. Is that true or false? I hope you were listening. Mycoplasma. They have a waxy wall. They have a thick waxy wall. Hope you were paying attention. Now we're gonna see who's paying attention. It's Deontay, right?
Yes.
Okay, so like you just said. All right, this one's false. They don't have any wall. Deontay called that one out. They don't have any wall. That actually were the next person I was going to call, but since you said that thing, we are covered. All right, so they don't have any cell wall at all. So they are gram negative. Actually, they'll stain gram negative. Stain will be red because we don't have peptidoglycan to trap the purple in there. That's why. So they'll stain red, but they don't have any wall at all. Mycoplasma. Mycobacterium, acid fast, and they have a thick wall with that wax on it. Yep. They're gram positive. It's already something we're learning about here. All right, next question, question 6. What are bacilli? That's an easy one. Come on now. What's bacilli? There's really two major shapes that I want you to focus on because those are the only two shapes I have in the lab. So if you know the two basic shapes, you'll be good. And those are cocci and bacilli. You know one, you should be able to figure out the other one. So bacilli is rod shaped. And this is John, right? All right, what's round shape? What's the word for round? Coccus, excellent. Coccus is the round shape. Bacillus is the rod. Bacilli is multiple. Cocci is multiple. So it's just plural versus singular. Okay. Corkscrew, we might call those like spirochetes, spiral shaped. Spherical bacteria that grow in pairs. So spherical bacteria would be cocci. If they're in pairs, we would call them diplococci. In fact, Neisseria meningitidis is a diplococcus. All right, moving on. You guys are doing great. We're almost there. All right, what does peritrichus mean? In case you can't tell, it has to do with flagella. So look up in your notes where you have about flagella. Peritrichus. Alrighty. Peritrichus is flagella distributed over the entire cell. Kylie. Is Kylie here? No. Oh, okay. Is it lip or lippa? Lippy. Okay. All right, I'm not going to remember, but you know. All right, lippy. So Kylie, can you tell me the word for when we have a tuft of flagella at one end?
Say it.
The L one. Lophotrichus.
Yeah.
So the 4 arrangements we can have, if you have a flagella, you are motile. The flagella is for motility and is the only appendage bacteria have for motility. There's no other way they can move around. The pili are for basically exchanging information typically. Fibrae are for attachment. Bacteria don't have cilia, right? And they don't move like amoebas do, but that pseudopod motion. We'll learn about that in chapter 5. That's for the eukaryotes only. So if you are a bacteria and you are motile, you can move around, you have a flagella, at least one. Okay, if you have one flagella, that is monotrichous. Like some of us probably are trichotillomania. Trichotillomania is where they pull the hair out, right? So trick means like trichology is hair. Okay, so these are hair-like appendages. And they move in a rotating fashion. They do not move in a whip-like fashion. So they either rotate clockwise or counterclockwise. And based on if they're rotating clockwise, they'll do a run. And if they suddenly switch, that's a tumble. And that's how it's not very scientific, right? It's hard to control how they're moving. They'll just run and then tumble until they're going in a general direction toward or away from something. And that movement, we call it taxis. If you're moving to a chemical, like let's say I had some cookies up here. Y'all would smell that smell and probably want to come eat the cookies. That's chemotaxis. You're responding to a chemical in the air that you're smelling. If I had a bag of poop up here, you'd want to move away from it. It's still chemotaxis, but you're moving away from a chemical. And bacteria can do that too. Sense how close or how far they are from a chemical signal and move towards it just by running and tumbling. All right. Eukaryotic cells, on the other hand, they have whip-like flagella that are covered by a whole membrane. And so they have a much more controlled movement. Of course, they've got a lot more complicated cells too. So they can control that stuff more easily. But very basic stuff with our bacteria. But you can see now, if we're saying all you can do is rotate one way or another way and run and tumble, if you have a whole bunch of flagella all around you, rotate some of them on one side and some of them on another side. And now you have a little bit more control over how you're moving. All right, so our peritrichous guys are covered all over. Monotrichous has just one flagella. That's it. It should be at one end or another. A tuft of flagella is called lophotrichous, and amphitrichous is having flagella at each end. We're talking about ends of the cell. We're typically talking about our bacilli, right? Cocci typically don't have flagella, but that's not 100% always the case. It's hard to make hard and fast rules with bacteria. Okay? Yes.
So the one flagella would be like a sperm cell?
Sort of like a sperm cell, yeah. So that would be like what we could say monotrichous, but for a eukaryotic cell. Yep.
Is that if most sperms have is like a
Yes.
okay.
Yeah. So we can actually think of that as our base understanding of a eukaryotic flagella, because we know that sperm do that whip-like motion. They can wiggle that tail all around. But our bacteria literally just spin it one way or another. If you're looking at it, it might look like it's whipping, but they can't control the whipping. It's just being moved by the elements around it, if you will. You will never see flagella, by the way. They're too small for us to be able to see on a microscope. So when we look for motility in our bacteria, we'll just look to see if they're moving or not. If they are moving, they have flagella. We won't get to see their arrangement. There are special stains we could do to look at the flagella because they're too small to see with just regular staining. They're too small to see without staining, which is how we look at the motility. So we won't look at our actual flagella, but it's actually one characteristic we can use to know different species of bacteria. For example, Salmonella has peritrichous. It has flagella all over it, whereas E. coli is typically lophotrichous, has a tuft at one end. So that's even one structural characteristic that we could use to compare the two. They're both gram-negative. They both live in the gut, but it's different. All right. Next question, question 8. The bacterial capsule. Tell me about it. This is an image of some bacterial diplococci with capsules around them. Or diplobacilli, I don't know, sort of like rod shaped. Definitely diplo. What's the capsule do? All righty. So capsules, they're a sticky coating that the bacteria might have, and that your cells actually have, by the way. It's a sticky coating, usually made of sugars or protein, or a mixture of sugars and protein. Usually sugars, and it's sticky, like honey. Once densely associated, we call the gut sticky layer the glycocalyx. Okay. Glyco, again, sugar, like we had with peptidoglycan, like glyco sugar. Okay. So the glycocalyx is a sugary outer sticky coating. If it's very loosely associated, you're nice and gooey and yucky, then that is a slime layer. But if it's densely associated, like we saw in the picture on this, very tightly associated, very clear and defined capsule here. then that is what we call a capsule. It's just type of glycocalyx. You're either a slime layer or you're a capsule. These are capsules. Sugary for attaching to surfaces, for building biofilms, which are when layers and layers of cells kind of stick to each other and create a little community. Okay. Or for evading phagocytosis, which was our answer here. So anybody know what phagocytosis is? Yeah.
Isn't that done by your white blood cells and it kind of just like destroys?
Yes.
things?
So yes, so phago is, if you guys have taken med term, then you might know some of this stuff, okay? Phago means to eat. Cytosis is to cell. So it's cell eating. So what it is when your white blood cells eat up the bad guys. So your basic function of a lot of your white blood cells, your macrophages, your neutrophils, they go around and eat up just what, like cellular debris, waste, and foreign material. Okay, so that's a process that we call phagocytosis, cell eating. So this sticky layer, if I were to coat you in a thick old layer that's twice as thick as you are with some honey, you'd be slipping away from the cops when you're trying to rob a bank. If you had a thick old layer twice as thick as you of sticky sugar on you, it would help you stick where you're going, but also be hard for the cops to grab onto you. It's the same thing in your body. Your white blood cells can't grab on when there is a sticky capsule there. So that's one of the functions of the capsule. Evading phagocytosis, protects against our white blood cells eating the bacteria. And I hate to break it to you guys, most bacteria have either a slime layer or a capsule. And so it's very easy for them to evade phagocytosis, but don't you worry. Your body has ways of getting around that, which we'll learn about in Unit 3. Okay. So it's made up of sugar, not of lipids. It does not participate in the telophase and mitosis because, I also hate to break it to you, bacteria don't go through mitosis or meiosis. That's a eukaryotic thing. So bacteria, when they multiply, this is in chapter 9 thing, but they go through a process that we call binary fission. It's different than mitosis. Okay. They don't have to make the multiple copies of the chromosomes and split them apart because they've only got one chromosome. That's really the key is in mitosis, right? Remember PMAT. I don't know if you guys ever learned about that stuff. Prophase, metaphase, anaphase, and telophase, which is in chapter 5. Sorry. But you next time. We'll get you on that next time, okay? And not composed of mycolic acid. That's a feature that we only see on our mycobacterium. That's on their wall. It's not a separate thing. The capsule is a separate thing. Could a bacteria have mycolic acid in their wall and have a capsule? Absolutely. But they're two separate things. Okay. Right. In fact, Mycobacterium tuberculosis does have a capsule. All righty. I didn't ask a question on that one. Sorry. Break your little hearts. Next question. When are endospores produced? Got Kylie here. Other London Cooper. Okay. Sorry. You'll be the next person. And Kate, Brandy, Saber are not here, correct? Kate Henderson? If you don't speak up now, I'm giving you 0. Okay. Okay. She hasn't dropped it yet if she isn't, because I checked it before class started. All right, so when are endospores produced? All right, our answer here is in nutrient deficient conditions. Now, a lot of us picked moist, warm conditions. Interesting. So I'm going to call on London. Tell me about what the purpose is of an endospore. What are they? General. Facilitate survival, right? And so these are things that we will produce in usually nutrient deficient or dry conditions. They're harsh conditions, right? So endospores are only made when we need to hunker down. Think of it as like armadillos when they ball up in the armor to like hide away from their threat. It's sort of like that, but it's like a whole metabolic thing. So a normal, happy, multiplying bacterial cell, we call that a vegetative cell. Okay? And some bacterial species, in fact, only 2 genera here. So yeah, you better believe I'm going to test you on which genera are acid fast and which genera are endospore forming. Okay, so our acid fast one, Mycobacterium, right? are endospore forming ones. Bacillus, it'll be in the name, bacillus. Now, I'm not talking about their shape, I'm talking about their name. Bacillus will be in their name, okay? Are they also shaped like a bacillus? Yes. But are all bacillus-shaped, rod-shaped bacteria called bacillus? No, like Escherichia coli, not in its name what shape it is. If it's in the name, bacillus is in the name, it's an endospore producer. Okay. The other one, the name is Clostridium. Clostridium. So bacillus or Clostridium are in the name, and they are both gram-positive. Okay, that's key for what our activity is next. So remember that endospore-producing bacteria are gram-positive. So if I end up giving you a test question, and I'm like, this person has endospores in their sample. When the lab tests them, they're gram-positive. Does that track? We will be more eloquently written than that. You should be able to tell me whether or not gram-positive tracks. Does, right? Okay. So gram-positives produce endospores. Not all gram-positives, only if you are Bacillus or Clostridium, but they're both gram-positives. If you're gram-negative, you automatically know you don't make an endospore. We can rule that out. In fact, we won't even test our gram-negatives in lab with the endospore stain when we do it. There's no point. It won't, if they're negative. They don't make endospores. All right. So endospores are only produced in very harsh conditions. Moist and warm. Does that sound harsh to you guys? To most bacteria, that sounds pretty pleasant, right? Most bacteria will like that. Moist and warm, that's not a harsh condition. So what state will the cells mostly like be in the moist, warm conditions? The vegetative, right? All right. The active, multiplying, toxin-producing, disease-causing form of these guys are actually causing the infection, will be the vegetative form. But usually the endospore form is the one that got in. The endospore got in, and then the moisture and the warmth and the nutrients woke it up and then turned it into vegetative. This is a cycle that it will switch between. It is not a reproductive cycle either. One vegetative cell makes one endospore. One endospore makes one vegetative cell. You can cycle between the two, but it's not reproductive. It's not a way to multiply. It's just a way of hunkering down and waiting out things. These are extremely medically relevant. Clostridium botulinum, what do you think it causes? Botulism. Right? Clostridium tetani, tetanus. Clostridium difficile, C. diff. Clostridium perfringens, you might not know this one. Gas gangrene. Gangrene. Okay. So there's 4 right there. All right, our bacillus guys, less common to cause illness. Most of them are actually good friendly gut bacteria, but one of our disease-causing bacilli is Bacillus anthracis. What do you think it causes? Anthrax. Okay, and there's a difference between these two groups too, drastically different. Bacillus species typically need oxygen to live, but Clostridium species, they will die in the presence of oxygen. So that's a key thing for C. diff, because when it's outside of the body, it turns into an endospore. It's like, but you get it into the body, and once it's in that anaerobic, there's no oxygen inside of your body. Like in your gut, there's no oxygen. In your stomach, there's no oxygen. But you can ingest C. diff pretty easily. The endospore will survive your stomach acid very, very easily. Endospores are so hardy, they're super difficult to get rid of. So they'll survive your stomach acid, your mouth, saliva, everything. And they get into your gut where there's no oxygen, there's no acid, and they're like, finally, and they grow. You can have 1 or 2 of these guys just fine in your normal gut flora. But once those stadium seats are cleared out, have I gone over the stadium seat analogy with you guys? Think of your whole body as a stadium, and every tissue in your body is a stadium. There's so many seats in the stadium, right? Normal bacteria on your skin take up like the stadium seats in your skin. Your gut bacteria take up the stadium seats. There's less places for the bad guys to sit down. So they actually help out with preventing bad guy infections because the bad guys can't sit down in a seat. Your blood and your nervous system are like stadiums that are completely open. We don't have normal bacteria there to take up the seats. So that's why those types of infections can go very, very quickly and multiply and cause severe issues, because we shouldn't have any bacteria taking up those stadiums. And it's still an open stadium. It's also, they're closed systems for a reason. So things aren't supposed to be able to get in and out. These are very crafty pathogens, so they can get in and out. So In the gut, your stadium seats should be taken up by your normal gut bacteria. In fact, in 1 centimeter of your colon, there's more bacteria in 1 centimeter of your colon than there have ever been people on the entire planet in all of history, forever and ever and always. In 1 centimeter of your colon right now, there's more bacteria than have ever been people on the planet. Isn't that wild? So anyways, so they're taking up stadium seats in your colon, but you take antibiotics and you're opening up stadium seats in your colon. And now the C. diff, where you might have had like 1 or 2 seats taken by C. diff. C. diff hunkered down and turned into an endospore while you were on the antibiotics. When you were off the antibiotics, C. diff was like, and just like populated all those open seats. And now you've got pseudomembranous colitis, which is the disease that's associated with C. diff. All right, so that's the idea there. We'll learn about that. That is a concept called microbial antagonism, where our own microbes are protecting us. Yes.
Like in the hospital, when you have C. diff, they say like infection control say 6 months
Mhm.
before you can take them off of the C. diff precaution.
Mhm.
Why would they say 6, like,
Yeah, so we know like a lot of our other bacteria, whenever you take antibiotics, it's like 10 days of antibiotic. That does not apply to all bacteria. C. diff is incredibly difficult to kill. I mean, very seriously, very difficult to kill. Probably as difficult as tuberculosis. But C. diff, the reason it's hard to kill is because it turns into an endospore. So what it'll do is the living part of the cell is what's killed by antibiotics. Because antibiotics usually interrupt some sort of cell division thing. We inhibit it from making a cell wall. Well, if I'm an endospore, I don't need to make a cell wall. You know what I mean? And so those endospores can live for decades hunkered down. And so like when we talk about anthrax endospores can live in the soil for like decades and decades and decades. And then a farmer goes and plows the field and the spores get into their lung and it turns into the vegetative form and leads to pulmonary anthrax.
so the antibiotic is not necessarily,
So it'll kill off the living version of it. And the idea is if we can keep on the antibiotic, then eventually we can outlive the endospore, right? And so if they're not populating your gut, if we're on antibiotics that target C. diff, but not your other bacteria in your gut, then we can keep that endospore in endospore form. And hopefully the other good bacteria will overgrow and take up the stadium seats. You get what I'm saying? Because the endospore isn't taking up a stadium seat per se. But the other bacteria that need the nutrients can hopefully regrow.
You know, like crime shows when they're talking about like bioterrorism, are they using like
Endospore.
endospores?
So, and that's why anthrax is so well known to be a bioterrorism agent. You just get some endospores of anthrax that are a little bit powdery. They're a white powder substance. You can put them in the envelopes. Like there was an incident of that with Congress, like in 2002, I think. They mailed several letters to Congress that had anthrax in them. And so they had to like, basically fumigate the whole building because of that. And I don't think anybody actually got anthrax from it, luckily. It's not like super easy to get it, but it is also, once you get it, we're talking the best form of anthrax that you can get is the cutaneous form. And it's like the skin version. So you have an open wound and the spores get in there. Yeah, and it turns black. We call it an eschar. It's a black wound. Anyways, so you get that type of anthrax and it's 20 to 30% fatality rate. And that's the best kind that you can get. I mean, that's like your best case scenario. So you get pulmonary anthrax or GI anthrax or something like that, then we're looking at 50 to 70% mortality rate. So yeah, somebody was breathing those spores in and they actually got anthrax. It's really high rate. It's not a good way to go either. Anyways, any other questions? All right. How would you describe the strepto arrangement? All righty. Kaya, I'm going to come back to you on this one. This is your last chance to get your points, okay? You need to look up in your notes, Strepto. There are a couple of them that you need to know. All right, so strepto. I think of strepto sounding like strip. Strep sounds like strip, and a strip like a strip. It's like a chain. Okay. All right, staphylo. Kaya, what's staphylo? Do you remember? If you don't know this one, you're not getting full credit today. Yeah. You know what to do if you don't get full credit? You can still email me if you guys, this happens to you. If it happens to you and I call in and you don't get full credit, I will let you know. But you can always email me the question that I asked you and what the right answer is to it. But you have to remember your question and I will not prompt you to do it. You have to do it on your own. Okay? So you can still get full credit, but you'll have to email me what my question was and what the right answer was. Okay? So, would anybody here know what staphylo was? Staphylo.
That's the
The clusters. Yeah. Staphylo is the clusters. Strepto is the chain. What's pairs? Do you know pairs, Kaya? Diplo. Right, we know that one earlier. Okay. And the spiral ones have a whole bunch of different names they could be. All right. Okay. All righty. There is one other arrangement that only applies to bacilli. It's called palisades. Anybody know what that one is?
Where instead of they're going like this, they're like this.
Like a fence post. Yeah, they're upright next to each other. Only the bacilli can do that for obvious reasons, right? But the roundwise, you can't tell. They're just strapped up. You can have the bacilli, the rod-shaped guys, be end to end, and that's still strepto. But when they're upright next to each other like fence posts, that's called palisade. Okay. All right. Here's our podium, and then we will do really quick our little worksheet for this one. We got 10 minutes left. Any questions about that stuff before we go into our worksheet? All right. And Kaya, if you know any of these things, you can save yourself with the worksheet stuff too. Okay. Remember, I'm going to call on all of you every single day. So be ready for this every day, every Monday. We'll do this. All righty. Our worksheet for chapter 4, we have a case study. And so I normally would give you guys a little time to look over this before we go over the question. I'm going to walk you through it since we have 10 minutes left. It does talk a little bit, obviously we're talking about anthrax here. So we know anthrax, which I talked about earlier, it's Bacillus anthracis. We can see here, Bacillus anthracis is the name of the pathogen, okay? We have anthrax, talks a little bit about human disease with anthrax, and then the actual study starts over here. We got Bacillus isolated from a patient that had pneumonia. So they had a lung infection with what ended up being the anthracis, which we found by nucleic acid amplification and gamma phage, which we haven't learned about phages yet, so you might not know, but nucleic amplification, we are talking about PCR. Okay. We haven't learned about the phages yet. That's chapter 7, so no worries on that. And then whatever. So we found that this person had anthrax, which is associated with the endospores, which we often see in farming areas. This patient was a 61-year-old Florida resident. They went on a 3-week trip in July of 2011. They drove through North Dakota, Montana, Wyoming, and South Dakota, which we can agree is a heavy farming area. And so this is an area where we have animals that have anthrax endemically in the animal population. So when we have an animal population having a disease, we call that a zoonosis. When we call something endemic, that means that it's naturally found in that area at a normal level. Okay. All right. So we aren't concerned about bioterrorism being the threat here. They were in an area where we found a lot of anthrax naturally in the animal population. All right. So let's go down to our questions and start looking at our questions. Delete that. Look at our questions. What is the disease discussed in the case study? You know this one, don't you? Well, what disease were we just talking about? Kaya. Anthrax, right? Okay, anthrax is the disease. All right, what is the scientific name of the pathogen? Bacillus anthracis. Okay, the pathogen that causes the disease discussed, which we know is Bacillus anthracis, is a bacteria. And is the bacteria gram-positive or gram-negative? It's not in the case study, but we should know this. We talked about it just a minute ago. It's gram-positive. Well, how do we know that? Because it's bacillus, it makes endospores, right? If it's endospore forming, the only ones that can cause endospores are bacillus species and Clostridium species, and they are both gram-positive. What does gram-positive mean? Thick wall. Right, so we're thinking about that. All right, does the pathogen discussed in the accompanying case, oh yeah, so it says, how would you describe it? Cellular envelope, thick cell wall. No, is there an outer membrane? Nope, only gram-negatives have that. Does the pathogen discussed in the accompanying case study have a nucleus? No. How do we know that? It's a bacteria. Yeah, it's an eukaryote, yes. So all bacteria are eukaryotes, which are no nucleus. No nucleus. What type of biological molecule is the genome made of? Nucleic acid. What particular kind of nucleic acid is the genome? Starts with a D. DNA, right? All bacteria, any cell, if you're any kind of cell, I don't care if you're eukaryotic or eukaryotic. If you're a cell, you have double-stranded DNA as your genome, okay? DNA. The pathogen discussed in the case study generates endospores. What is an endospore? Survival, hardy, dormant structure. They do not multiply. They are just dormant. They're completely inert. Just there to hunker down and wait out the harsh conditions. And man, do they hunker. And they can resist even disinfectants and things like that. The only way to get rid of them is through sterilization practices. That's incineration and autoclave. Our disinfectants that we use on our bench tops, they don't get rid of endospores. 99% of the time, if something is a disinfectant, the thing it doesn't get rid of is endospores. Okay. All righty. The pathogen discussed, it generates endospores when it causes disease. Are the endospores producing the toxin that it makes the disease with? No. Why is that? The vegetative state makes the toxins. Because when we talk about endospores being dormant, we mean that quite literally. They are inert. They are dormant. They are not doing anything. Okay. All right. Everybody good on those answers? Everybody tracking on why the answers are what they are? Now is the time to ask questions. All right. It is 10:46. Lab is at 11. I will see you guys up there in a little bit. That's it for today.